Good afternoon, everyone. My name is Gabriel Heckerman, and this summer I worked within the lab of Dr. Sean Wu with Dr. So Lee, focusing on hypertrophic cardiomyopathy, specifically modeling of the G256E mutation. Just a little background on the issue. Heart disease is the leading cause of death worldwide, and currently roughly 48% of US adults suffer from some sort of heart disease. Within that realm, hypertrophic cardiomyopathy, or HCM, is the most common form of inherited heart disease. It affects roughly 1 in 200 individuals, and it is a leading cause of sudden cardiac death, which essentially causes many young, apparently healthy individuals to just drop dead out of the blue. As you can see here by this graphic, HCM is a disease characterized by abnormal thickening or hypertrophy of the heart muscle. On the right here is a heart suffering from HCM, and on the left is a normal heart. And in the past, HCM has been linked to 1,000+ mutations, roughly a third of those being in beta-myosin heavy chain, which essentially on a molecular level is the primary human ventricular motor protein coded by the MYH7 gene. An important thing to notice here is that biological relevance of in vitro MYH7 mutation modeling, or essentially just modeling in a petri dish, has yet to be fully established. This leads us to the question that if the G256E mutation of the MYH7 gene is introduced into cardiomyocytes, or the contractile cell of the heart, and compared directly to a very similar or isogenic control, would the resulting transcriptome reveal biologically relevant recapitulation of disease cells? So essentially, we're looking at trying to create a relevant model of what this disease would look like inside of a human body. To do this, we used a tool called CRISPR-Cas9 for gene editing and introduction of the G256E mutation into healthy induced pluripotent stem cells. From there, we cultured these human-induced pluripotent stem cells and differentiated them into cardiomyocytes. With this, we allowed the cardiomyocytes to mature to day 30 and perform single-cell transcriptomic profiling. Finally, we used R coding language for single-cell RNA sequence analysis and manipulation of the data set and model comparison between G256E mutant and isogenic controlled samples. To begin, I started by just confirming that these samples were indeed cardiomyocytes. So I used known canonical markers, or genes that are known to be highly expressed within cardiomyocytes. I chose 2-titin and phospholambin. And as you can see here, both very highly expressed in the samples of the G256E mutant and isogenic control, each dot representing a cell. The same thing is shown here, just a map of clustering, each dot representing a sample, and the bright purple representing that is highly expressed across all of the samples. Basically just confirming that these are indeed cardiomyocytes. Into the bulk of our results, I have this heat map showing the G256E mutant batch and isogenic control. Down the left here are all the genes tested, and each little block here across this line, all the way through, are samples. Now, like I said, this is the G256E mutant batch itself, and what stands out is the variability of very high expression here, shown by the bright yellow, and very low expression here of the same genes, just in different samples within the G256E mutant batch. What we can draw from this is that there is variability of gene expression within the G256E mutant batch itself that has nothing to do with the batch being mutant versus the isogenic control. The conclusion we drew from that is that the current cell culture condition was not sufficient to produce a biologically relevant in vitro disease model. However, the benefits of this knowledge is that it provides a deeper understanding of the methodology used for this G256E mutation model, which is critical for determining the next steps in investigating molecular pathogenesis of HCM using in vitro models. Our next steps would be to evaluate the influence of cell culture condition on biological relevance of this specific model. Some factors we might include would be stressors, diet, exercise, or even method of mutation introduction. That concludes my presentation here today, but I would like to include some acknowledgements. Of course, Dr. So Lee and Dr. Sean Wu, Stanford CVI faculty, Adrian, Yemeni, David, and last but not least, all Stanford Cardiovascular Institute speakers for providing me invaluable knowledge and experience this summer. Thank you.